Oh, yeah.
It does that.
Yeah.
Okay. Okay. Awesome.
All right. So I'll hand it off to Rachel. Uh,
All right, thank you so much, Matthias. I'm going to share my screen. You can see the slides, everything look good? Yeah, okay. See some nodding. Great. So hi, everyone. Thank you for joining us. It's a pleasure to be here to share a little bit about what the journey to be a PI is like. So I have just a couple slides prepared to give you the highlights about what I did before I made it to UCLA, which is shown here. So I am a developmental biologist, and I think the question that I've been really interested in and excited about for a long time is how do plants continue growing and produce all of these new organs after embryogenesis? So how are they able to create post-embryonic organs? To study that question initially, when I was a graduate student, I went to the University of Maryland for my PhD, and I joined Zhangqi Lu's lab there. And I chose to do that because she was working on what to me was an interesting model, which is shown here. This is Fragaria vesca, which is a diploid strawberry. So I combined my interest in systems biology and also in developmental biology to study the transcriptional regulation of flower and fruit developments in strawberry. But so I finished my PhD here in 2018. Here's me, very happy with Zhangqi, my advisor. And immediately after graduation, I moved from the DC area to North Carolina, to Durham, where I started my postdoc at Duke University with Philip Benfey. And I chose to join the Benfey lab because kind of towards the end of my PhD, I began realizing the questions I was really excited about were fundamental biological questions, fundamental developmental questions, like how does a cell acquire its identity or how does a cell differentiate? So I chose Philip's lab because I wanted to use the Arabidopsis root as a model system for that question. The reason for that is because it's very easy to trace cell lineages in Arabidopsis roots. So you've got your stem cell niche here at the tip, and since, of course, cells don't move in the root, you can see the whole developmental trajectory, the whole lineage, like this one shown here with GFP, as you move towards the shoot. So for my first major project in Philip's lab as a postdoc, I generated this single-cell RNA-seq atlas of the Arabidopsis root, which is shown here in this UMAP plot where every dot corresponds to an individual cell, and they're colored here according to their cell type. So we generated data both for the wild-type root and also applied single-cell RNA sequencing to developmental mutants. After this was published, kind of for the rest of my postdoc, I was very lucky to have a chance to contribute to a lot of different projects, both thinking about how to apply single-cell RNA sequencing technologies to plant tissues and how to use this technology to address questions in developmental biology. So after my postdoc here, I transitioned and came to UCLA. We were just talking at the beginning about driving cross country. So I drove cross country in March of last year and arrived at UCLA for my very first day on March 1st of 2024. And since then, I have been extremely lucky. I think community is a really critical part of starting your labs and UPI. Very lucky to have started with an amazing team in the lab of postdocs and students, and also to have a great environment at UCLA. So my lab is right next to Jamie Van Norman's lab. Here's some representative lab members here. And also we're right next to Steve Jacobson's lab. So I think I'm very happy at UCLA and very happy with the community that we have built and are building here. So in my new lab, we're addressing two major topics that are launched with projects I began while I was still a postdoc. So the first one, I'm really curious about this question: how do root transcriptomes change as the seedling ages? So all of my work previously has been in these 5-day post-stratification Arabidopsis roots, and I just wondered what happens over time. Are there cell fate or cell state changes, and can we address that by looking at the transcriptomes? So similarly to the atlas, I generated single-cell RNA sequencing data from these root tips from all of the different ages, essentially, that you just saw in the previous image. So it turns out when we combine all the data, there's a lot of changes over time. So we're investigating what's going on in the lab and what's changing with seedling age. I focused a lot of time also on primary roots. So going back to this image I showed you at the beginning, I'm also now interested in what's going on with lateral root development. So we have some projects now stemming from things that I started in Philip's lab, thinking about gene regulatory networks and cell-cell signaling that are controlling lateral root initiation and patterning. So with that, for the rest of the panel, I think the one takeaway I wanted to tell you from my experience is that as a new PI, mentorship is really critical. So I'm really grateful for the peer mentors and also more senior mentors I've met through various venues, Plant Cell Atlas, the Leading Edge Symposium, of course, my department, and the plant biology community. So I'm happy to answer your questions.
Great, thank you, Rachel. We will move on to Kevin and have him tell us a little bit, yeah, about his experience starting his lab.
Sure,
And
let me get my,
yeah, sorry.
no, it's all good. Let me get my slides pulled up. Okay, and then jump back here. Okay, and you guys can see that? Awesome, cool, see now. Cool, yeah, so thanks for the invitation to those that organize this webinar series. So I'll share like a little bit about like my journey as a PI and then like I guess kind of what I've done to kind of get to that point. And so I kind of start with like this question, like how did I actually get into science? And so I kind of start with like, I started with going into biology through getting a bachelor's at the University of Missouri in St. Louis. And at the time I was going there to become a pediatrician. And what kind of actually changed my career trajectory is I took a microbiology course and I got really fascinated by studying like microbes and how they harm us, how they help us, and how they do all these cool things to all these other organisms. And so that ultimately diverted my interest into going to research. Now, the problem with that at the time was like when I decided to go into microbiology, I was still working at this place called a dollar store as like a sales associate. And I had decided at the time, well, maybe I should start getting some lab experience if I actually want to go into the lab to see if it's good for me. Um, and so that's why I kind of got my first exposure to the Danforth Center to where I got a part-time lab position to be like a lab assistant and get my first exposure to plants. And at that time, I worked in Todd Mockler's lab, who passed away a few years ago. But in his lab, I worked on the brachypodium transformation pipeline and got really hands-on experience as far as like tissue culture, plant phenotyping, like plant genetics. It was really my eye-opening experience because before then I really could care less about plants. And this was like the first time I got really exposed and really excited about plants. And that kind of took me off to grad school. So I went to grad school at Texas A&M, got advised by Lebo Shen studying like plant microbe interactions. And with that, I basically studied how does this bacterial disease on cotton, what are the molecular mechanisms that cause this disease? And what my project involved was looking at this sugar transporter called Sweet 10 and pretty much identifying it as a direct target to cause this like bacterial blight disease system. So it was really cool because I was like one of the first ones to identify this molecular mechanism, and that kind of helped fuel my passion to kind of go into like basic science and study molecular mechanisms and how plants respond against like pathogens or stress responses at a transcriptomic level. So for my PhD, I kind of focused on like plant molecular biology and functional genomics, and I kind of collaborated with other groups on the transcriptomics aspect of my project, and that was something I really wanted to gain more experience in. And so I went to a conference during my PhD in China. It was about back in 2016, I believe. And when I was at this conference, I was fortunate to bump into my former postdoc advisor, Blake Myers. And we had like a great conversation. We followed up with each other and ultimately ended up with a job. And so I guess my advice with that is like, do not be afraid of network as you're kind of going on through your career, whether you're an undergrad or grad students or postdoc. Networking, it does get you a long way. So I kind of advise that to you all. So I had joined Blake's lab in 2019. So I went back to the Danforth Center. And there I specialized in spatial genomics and I also worked on duckweed biology. And that's where I kind of got really excited about duckweed early on. And I'm also like a current like 2019 Henry Gray fellow. So I have this postdoctoral fellowship that kind of funded my postdoc and then also helps me start up a lab like in my early years as a PI. And so how I ended up in this current role is a long twisted story. So, like I said, I got my PhD at Texas A&M. Then I did my postdoc at the Danforth Center, studying all these different methods and doing these different techniques. Went on the job market, which was grueling, but eventually got through it. And I ultimately ended up with a great position to be a joint faculty between WashU and the Danforth Center. And July 1, that's why I started my faculty appointment. So I'm coming up on roughly 1 year of my faculty appointment. And it's been a chaotic fun, which I like to phrase it. If I could express my experience in a few words. And I think lastly, like the reason why I'll share a little bit like why I wanted to join appointment because it's a little different for like a junior faculty. So with WashU, I kind of envisioned myself like I really want to get into like mentoring, training, like and teaching undergrads and also recruiting grad students and postdocs. And then I also saw my science as very interdisciplinary, so I could collaborate within different departments such as like engineering, physics, chemistry, and really like work together on like projects. At the Danforth Center, it's a very large population of plant science, collaborate with a ton of different plant biologists there in one concentrated like building, and then also like outreach to the St. Louis region. I'm a St. Louis native, by the way, so I was born and raised in St. Louis. So giving back to my community, like, is very, it's a passion of mine. And then ultimately, like, it kind of serves to strengthen the collaboration and kind of serves as a bridge between these two institutions. And to share a little bit of what my lab, what we're doing right now is we're basically trying to understand how do plant cells regulate, communicate information. And we're kind of doing this at a transcriptomic level. So in order to understand like how our plant cells communicate and regulating information across organs or across the entire organism, you kind of need to understand like how our genes or proteins or transcripts or whatever biomolecules you're interested in, how they're being communicated with the spatial context. And so my lab is kind of like in two parts. So we go big on the basic or discovery science to understand like how do cells use gene expression to communicate information across a plant in the context of plant pathogen interactions and duckweed development. And I basically use the analogy of like you basically taking a pile of Legos and making a shape with it. And so that's why I kind of viewed spatial biology as like you're now putting these pieces together to make a three-dimensional shape to figure out like where are things like at with the spatial context. And then there's also a little bit of an applied / impact focus on my lab to where we kind of use duckweed to improve the human conditions. And so there's other things you can use duckweed as far as like, being good for biofuels or like food source or animal feed or phytoremediation. And so there's some stuff in my lab that we're kind of working on to go in that direction. But we're kind of more focused on the basis side at the moment, and then there's like a little bit of applied / impact focus to it. And so with that, I'm happy to have my lab. They make coming to work fun every day, even though despite the times that we're in, like they make coming to lab and mentoring in my job just fun. And so I really appreciate each and every one of them. And so with that, I'll take any questions towards the end of the panel. So thank you.
Great, thank you, Kevin. And now let's move on to our last panelist and hear about her trajectory, Monica Fisher.
Hello, everyone. I did not prepare a presentation. I was just gonna share my journey with you all verbally. So I have a little bit of a unique route into science. I actually started in the culinary industry. So I worked in kitchens all through high school and was really passionate about food and went to culinary school. And it was actually through food that I discovered science and biology. And I think looking back on it, I think I was always a bit of a scientist. I was a very nerdy chef. I really wanted to understand where my food and vegetables were coming from, how were they grown, how did that affect the way that they tasted and the way that they looked. And I had a really incredible internship at a farm-to-table restaurant in Washington, where I would work on the farm in the morning and harvest my vegetables, take them into the kitchen, prepare them, serve them, and then collect the waste and take it out to the compost. And it was really incredible to just be a part of that full cycle. And the farmers on that farm were the ones who taught me about the soil microbiome and mycorrhizal fungi. So I'm a fungal biologist, but I've always been very obsessed with this question of how are fungi interacting with other organisms in their environment. So it began with mycorrhizal fungi and how are fungi communicating with their partners? How are they communicating with the rest of the soil microbiome? I just got obsessed with all of these questions of fungal communication. And since I'd been out of school for some time at that point, I thought that like I could just like I can read, I can teach myself, like I can self-learn, I can go to my local mycological society and learn from the experts there. And there's a professor there who I was just constantly berating with questions. And at some point he told me, like, we don't know. Nobody knows. If you really need the answers to these questions, that's a PhD. Like, you need to go back to school and you need to figure out these questions. And I sort of took that as a challenge, as like, I'll show you. And so I think I was then maybe in kind of a unique position that I had this like crystal clear idea of what I wanted to do going into my undergrad. So I started out at community college and did all the prerequisites, and then eventually transferred to the University of Washington, Seattle. And I got into research labs as quickly as I could. I basically structured my whole undergrad degree around like, what do I need in order to answer these questions about fungal communication? So it's a lot of physiology, genetics, basic biology, some ecology. I'm very interested in like these molecular mechanisms that are mediating ecological processes that we see in the environment. So I had sort of a very general biology experience in undergrad. And then for grad school, I really wanted to focus on getting like mastering those genetic and molecular techniques. So I moved to UC Berkeley to do my PhD with Louise Glass, where I did a very genetics PhD on how two clonal fungal cells communicate with each other and solving that genetic pathway. Which was an awesome experience to really dive deep into the genetics. But at the end of that, I really wanted to be able to connect it back to ecology somehow. Like, why should we care that fungal cells communicate? Well, that communication process is necessary for the first step of colony establishment. And the fungus that I was working on is called Neurospora crassa, which is a wonderful model organism. But it's also a post-fire fungus. We see it fruiting from trees within days after fire. And we really don't know anything about what it's really doing in the environment. It's clearly establishing a colony and growing and thriving. But like, what is its metabolism? How is it being dispersed in post-fire environments? How is it interacting with other organisms? So that was something I was very interested to get at in my postdoc. And surprise, there was a lab just down the hall from me at Berkeley. There was actually a bacterial analytical chemistry lab that had a collaboration with a fungal ecologist, specifically studying post-fire fungal ecology. So I got to really kind of do my dream postdoc that then became the foundation of what is now my lab. And I think I was also in kind of a lucky situation that all the fungal biologists who I inherited the fungal side of that project from, they all retired, and my postdoc PI was a bacterial analytical chemist. Like, he didn't have any intention of continuing on my project. So my postdoc really was kind of an incubator for what is now my lab, and I'm able to bring all of that now with me to University of British Columbia, which that was like the most stressful move of my entire life, essentially moving a lab when like so many precious living strains in addition to my personal life and an international move. And it was wild. Also, starting a lab in BC in Canada is a little bit slower, I would say. We kind of have to apply for some of our startup funding. But as of a couple weeks ago, I just got my very first undergrad students, and my lab is finally like a functional lab. And we're kind of wrapping up some of the work from my postdoc work, looking at interactions between this cohort of fire-adapted fungi that thrive immediately after fire, and how sort of metabolism and nutrition is driving their interactions. Um, so I think I'm close to out of time or over time, but my quick 2 cents of advice for starting a lab is to identify and maybe stay focused on the sort of low-hanging fruit projects that are gonna get you papers, or like it's an easy thing to get at immediately, and it's gonna be an easy thing to mentor your people on, because I don't know. I'm curious to hear how the rest of you guys feel, but I feel like I'm being pulled in 25 million directions, and it's so important and grounding for me to feel like I have a clear idea of like, okay, this should be a simple project that we can just kick out, and it's always easier said than done, but I feel like having that intention is important. So, okay, I'm excited for the discussion and the questions.
Yeah, thanks, Monica. Um, we have prepared a set of general questions, and they kind of fall into two categories. One is like questions aimed at what is it like becoming a PI and that transition. And the second set of questions is like more of how is it to run a lab and more of the nitty gritty of that. And we thought first it'd be interesting to know what was, and some of you can have slightly answered it, but what was this ground or like this foundational motivation for choosing to become a PI, and when was that decision clear in your life? And I guess all you can pop in if anyone wants, like between the three panelists, you can answer.
I don't mind going first. So I originally wanted to go to vet school. I I had a horse in high school, that I leased and spent all my time at the barn doing chores, and it was really a very pleasant time. And I wanted to be a vet. And then I went to undergrad. Actually, I started in my hometown. There's a small university in my hometown, Salisbury University. My first year there, I learned what a PhD was. I didn't have anybody in my family who had gone to graduate school, never heard of a PhD. And I learned that you could get paid to do science. And so I decided I wanted to be a scientist and pursue intellectual curiosity. It was very, idyllic kind of mindset at that time. Turns out it's a lot harder to do that than I thought at the time. But that's what changed my mind, is actually meeting a scientist for the first time and getting some hands-on experience in the lab.
Yeah, I think with me, like I kind of when I started grad school, and just because like the experience I had like working in a undergrad in a lab as undergrad, like that's when I kind of said like, oh yeah, I think becoming a PI would be kind of cool. Um, I think it was kind of more firm when I was into my postdoc, and I like really was kind of more dead set as far as like, yeah, I really want to become a PI, and I really want to start my own program and mentor the next generation of scientists and everything. Um, because I think in grad school I was still kind of leaning towards PI, but I was kind of open to everything. But I think like the more I kind of talk to other people, like talk to my peers and other faculty to see like what it was like, that's why I kind of became like more dead set.
Yeah, I think for me, it was some point in undergrad or maybe grad school, and just finally understanding what academia is and like what it's like to be an academic, and that you have all of these different things that you can do. Like you can write, you can give lectures, you can teach, you can mentor, and also ultimately like the fungal biology community of people I utterly fell in love with. So I guess maybe to echo Rachel's point at the beginning, that it's all about community, that like at the end of the day I just want to be a part of this community forever and ever. And I've realized that this sort of style of it works really well for me. I love like part of me really loves being pulled in all different directions and having the ability to do a million different things. I find that really invigorating, and fulfilling. So, yeah.
Wonderful, thank you. um, we have this question prepared, and it's in the mindset that we as scientists are in these communities, and we exist as also lineages from the people who have mentored us and the people who have mentored them. And we were really curious about the people who have mentored you and what you uh, learned from them and what you think is important to bring over, if you think it's important to carry this over into your own lab?
I think something important that I took from my postdoc advisor, Philip, in my postdoc lab was how critical it is to have open communication in the lab. So he really taught our lab, everyone in it, to assume positive intentions about your colleagues, about the people in the lab with you. People really often mean to do the best, and sometimes there's miscommunication and that causes problems. But if you just sit down with people and talk to them about whatever's going on, whatever the issue is, sometimes that can help circumvent problems. So in thinking about what I learned from Philip and also some things I learned from HHMI programming that we were doing in our lab prior to the time when I left, shortly before I left, kind of thinking about how can you create a community feel in your lab and how can you ensure there's open communication among all the members and everyone feels welcome and able to contribute to the best of their ability. So that's something that's important to me to try to continue in my own lab.
Yeah, I think just thinking over my two advisors. So I think one big thing I took from Lebo was that I guess kind of like being really involved in like your trainees projects is kind of like fulfilling and kind of excites you in a way. Because like the one thing that she did a really good job was like she knew like her projects like of each of her trainees, and she had like a really big lab because she had shared it with another person. But she knew it like ins and outs, and because of that, she felt like she was a part of the project. She could help the trainees like with their projects, whatever struggles they were going through. Um, and so that's what I kind of do is like with my trainees right now is that like I make sure I stay involved with their projects and know them as try to know it as much as they do in a way, so I can kind of be a part of it as they are. And I think the big thing I took from I think one thing I took away from Blake was just the ideas and to just don't be afraid to think big with projects. Um, it's okay to get a little wild because sometimes it might be needed to actually push the needle in some direction. Um, so
Yeah, I think for me, a lot of I try to emulate my PhD advisor, Louise Glass. She was just such a calming, grounded, um, force. And I feel like I really learned a lot of scientific integrity from her. And as part of that, sort of taking the emotion out of science and being able to sort of separate yourself from the data and just trusting the data, trusting your science, and it doesn't reflect on you as a person. And at times it's like the most unexpected results that seem like you failed might actually be the most interesting. And so just making sure that I'm very clear about that with my trainees, and sort of emulating that and being a role model for scientific integrity, I think is very important for me. Um, and so for my postdoc advisor, so part of the reason I was excited to join his lab is because he started his lab about 2 years before I joined it. So he was a very new PI, and I was very curious to sort of witness firsthand what that process of starting a lab is like. And it was a little bit rocky. Um, and I definitely took some lessons from that. One of them is that he's very much a jack of all trades. Like he loves to do a bajillion different things. And I definitely see that in myself too. So I was very curious to see like, okay, how does this work? How can we make this work? And I think like it was a very exciting, interesting environment to be in, but it was also very disconnected. Like it didn't foster a sense of community because we were all working on completely different things. Like we were using different reagents, we were using different equipment. Like there was no reason for any of us to really interact. And so my lesson from that is to make sure that there is some thread that's connecting everyone in your lab. Maybe we're all using the same media or the same organism or answering a similar question, but something that sort of inherently connects everyone, I think is important for that community building aspect of a new lab.
Thank you. This is all very fun to hear, and it's always wonderful to hear like about everyone's experiences in so many different labs. Um, the next two questions are about kind of the mechanics of running a lab. So how did you guys approach recruiting slash staffing? And there's a second part to the question is like, is there any advice you would give your past self? And do you have any regrets on how you probably started a lab and maybe there was a better way to do it?
I'm sorry, Matthias, could you repeat the first part of the question? I got distracted thinking about my answer to the second part.
That's okay. It's how do you approach recruiting and kind of
Ah,
staffing, finding the right people?
right, right. So pretty much every mentor that I've ever had prepared me in advance in saying that recruiting the right people to your lab and the recruitment decisions that you make are the most critical thing that you're going to do as a new PI. So I came into thinking about this very hard, thinking about who do I want to have in my lab? What type of people? Who do I think I would work best with? I want to make sure that I'm not just recruiting a bunch of people who are just like me. How do I bring in people with different life experiences and different ideas? So I tried to be very conscious about that, and I would say I was very lucky. I try to be involved in my community a lot, in going and trying to give talks, and I try to say yes when I'm invited to speak on things like this panel, because it helps me to meet people and helps me to foster connections, I would say. So I was very lucky that some of my first recruits in the lab were people that I had already met prior to ever coming to UCLA, or people who knew other people, all in the same network. So I approached recruiting in that way, but through my network, I would say, through my community. Um, and the second part of the question was thinking about what we would do differently. Is that right, Matthias, potentially?
Yes.
Okay.
Advice to your past self.
Oh, advice to my past self, yeah. So maybe not about recruiting specifically. I'm thinking about this more generally. What advice would I have given to myself a year ago or 2 years ago? I think the advice I would give was to be a little bit kinder to myself, honestly. It's a really hard time to start a lab. It's hard to move your entire personal and professional life away from your postdoc environment, which for me was a very happy postdoc environment. It was also very difficult when my advisor, Philip, passed away 4 months before I left his lab. That was a very traumatic year and experience. So I would be kinder to myself. I think that I would have a little bit more confidence in myself that I can do this and listen to the mentors who told me that I can do it. Um, and just kind of enjoy the ride a little bit more than being so worried about doing everything perfectly.
Yeah, I think with me recruiting, I I guess first off, like I personally like having a lab size where like I feel like I'm comfortable giving my full mentor into somebody that needs it at any given moment. So that means not having a lab that's too big. So now like if you saw my lab size, we're pretty much like at a plateau because that's like my capacity right now to where I can feel like I can still give everything to everybody evenly. Um, and I think so that's one aspect. And another aspect is just like, what are like my projects do I need to push out? And then what skill sets do I need to push those projects? So if a project is like heavy on imaging, for example, then I should probably look for a postdoc or a potential postdoc that might be talented and passionate about microscopy and doing a lot of imaging. Maybe to the point to where like it surpasses like my knowledge because I have some imaging background, but like not to like a full heavy extent. Um, and I think as far as like trying to find the right people, I think with me, I've been listening very heavily to my gut, and that sounds silly, but it has been working. And I guess what I mean by that is if your gut tells you that it's not the right time to find somebody, or if it's not the right person, then listen to your gut. Um, I think that because I think it's like one advice I got that kind of took to heart was that like when you start a new lab, it's an empty space with no equipment or no nothing. And you have this like intention, like, oh, I gotta fill it, I gotta fill with people, and I gotta fill it fast. And you don't need to do that. It's very important, in my opinion, is that you find the right people, you find the right fits. And it's okay if you take a little bit of time to do that. Um, and so that's how I kind of gone about recruiting as far as like just taking my time, being patient, finding the right people, finding the right fits, and they've all been great uh, to this point. And I guess like what advice I would give to myself is actually, believe it or not, saying to Rachel is to give yourself some grace and just tell myself like you're doing a good job. Because it seems like that first year it's just like I'm just screwing up everything. Like you'll make a ton of mistakes like in that first year. You'll have doubts of whether like you're actually doing the right thing. Um, but you have to tell yourself, hey, you're doing a great job. Like this is a whole new role that you kind of have to figure out on a whim. So, but you're doing great.
Yeah, it's kind of the wild west of starting a new lab. But I agree that personnel are so important. Like that is your lab. And so like I'm taking it I think I'm thinking of that part of building my lab as the most serious thing, and I'm trying to be very intentional and very picky because these are the people who are gonna form the foundation of my lab, and it's gonna set the tone for who knows how many years forward. Um, and yeah, I've been putting a lot of thought and time and effort into that process, and like I don't know if it's a regret. I sort of wish I had figured it out sooner. I feel like I spent a lot of time kind of casually like having conversations with prospective students and not really realizing like, oh, that was an opportunity to interview them. And I should have had like sort of set questions that I'm asking everybody the same question. So like one for equity reasons, but also two, just to get a holistic understanding of who each person is. And there's definitely, I agree with Kevin, a like gut aspect to it. Um, and I've tried to be very explicit with prospective students about what I'm looking for and what skills I need, and that like I'm a brand new PI and this is gonna be crazy, and I'm looking for somebody who is like very self-motivated and can sort of work with me almost as an equal in a sense, to build our lab together. Um, so I think that takes sort of a unique type of person. I've also seen um, some of my fellow peers be the first grad students in other people's labs, and it's like it is a unique experience. And so I try to be really upfront about that. Um, yeah, and then screening for personality is something that I don't know. I don't know that there's like a good way to do it, but recently my mentor here said that one of her interview questions for prospective students is asking them to describe a time when they've been wronged and how do they deal with it. And so that like I love the question of like tell me about a time that like a science ex something you did scientifically like failed and then how did you deal with that failure? But I think it's also important to ask more broadly like how do you deal with being wronged, which also kind of gets back to Rachel's comments about communication and how do we deal with interpersonal conflict. And just, I don't know, I'm like trying to hire people who are both like scientifically, academically, like curiosity wise, like the right fit, but also going to contribute to a good lab culture. And then beyond that, recruiting, I feel like I haven't done much to recruit necessarily. I think by saying I study post-fire fungi, fire and fungi are like having a moment right now. And so if anything, I've been struggling to like filter through this like tidal wave of people that are like, fungi are cool, fire is cool. And I'm trying to respond to all of them and be like, I value you, thank you, but I need somebody with some sort of background in one of those things. So yeah, I think that answers the question.
Yeah, thank you all. It's been great. I'm gonna now transition over to Vincent, who's gonna help me moderate the public, Q&A section and any replies or remarks about your stories.
Oh yeah, thank you, Matthias. Thank you so much for your answers. They were amazing. It's really helpful to know how the managing of a new lab and how to recruit people and all these things are like important to continue with your own lab and how it started, how it's going and how it's gonna be. So we have a couple of questions. So I'm not sure if you already answered the Joanna's one in the chat. So she wants to know if you had any fear and what helped you with that fear. I'm gonna ask this directly to different persons. If you want to add something, please raise your hand like this, or raise your hand to in the Zoom call. So this question goes to Kevin. So if you had any fear, how did you manage that?
Yeah, I have here all the time. Um, I think the biggest thing is just that you have to, because the biggest fear is just like, I remember the first time where I hired my first person where it was my lab manager, and I was happy, but it was also like oh crap, I have a person I'm responsible for now. And like, oh shoot, what if I screw it up? And it's the same thing like as other people have come on, same as like I got my first two graduate students this year, so we're like, okay, well, now it's me that is they're kind of depend on me to make sure that I put them in the good best situation. And so I just kind of remind myself like, hey, like you were put in this position because people thought you were good and people thought you were like the right fit for the position. And I just tell myself like, hey, you were made for this. Like, just kind of give yourself a little confidence to kind of just lead. Um, I think it's okay to be scared, like as far as like this new role. But at some point, you have to kind of tell yourself like, okay, like they need me to lead, so I have to kind of get some courage and lead. Um, so that's what I've done. Maybe Rachel and Monica, if you got something to add, like feel free.
I have fear too all the time. I just solidarity with you, Kevin. I think that yeah, I just repeat my mantra that my mentors believed in me and they wouldn't have believed in me if they didn't, like they wouldn't have said it if they didn't believe it. They wouldn't have a reason to. So if they believe in me, I should believe in myself too.
Yeah, ditto. But also just talking to my colleagues here and my mentors here and being honest with them of like, am I screwing up? And everybody so far has been like, absolutely not. You're doing great. You're killing it. So, I mean, I think it helps to just share, commiserate.
Thank you so much for your answers. We have another question here. Luis wants to know if you guys still doing experiments, because we know that once you become a PI, they like focus on different things, more administrative, more like get trying to get funds. So he wants to know how you're doing with this. And I'm gonna start with Rachel.
Yeah, so this is a great question and one that I'm still trying to find the balance with, I would say. So I really tried to prioritize after hiring my first people, I really prioritized writing a grant, and which I'm still waiting to hear back about, fingers crossed. So I was really struggling at that time actually because I had never written like, a huge grant before. I had written fellowships and had fellowships throughout my entire career, but writing a full grant is a much different experience. And even working on like, with Philip, I would help him write, and with Zhangxi, my graduate student, my graduate advisor, I helped them write grants, right? But I wasn't responsible for coming up with every idea or the main bulk of the ideas or the scope. So it's a totally different thing to do that on your own. And so I was really in my office a lot and away from the lab a lot, and I was very unhappy. I was really miserable. So I received the advice that get back in the lab a little bit. You need to be in the lab a little bit or you're going to go insane. So I have tried now to balance with my rotation students, being more involved with them, obviously, and taking on more of their training myself. So that allows me to get back in the lab. And what I'm working on now is trying to identify this like a small something that I can do that can be my thing. That's okay if it gets interrupted, that I can work on to not only interact more with my lab members, right? I can't interact with them if I'm in my office. So I'm learning what's the best balance for me to allow them to have their space in the lab, but also to be involved with them there. This is really one of the hardest things that I've tried to encounter now. How do I split my time between being in my office and doing administrative things and learning how to do them while still staying in touch with the lab and in touch with people there? I don't think I have it all figured out, but I'm curious if Kevin or Monica have some other ideas on that.
Yeah, I kind of echo what Rachel said. I'm still trying to figure out that balance. I will say when I I had started somewhat my independent role from January up until like, I guess, July when I started my faculty appointment. And that's when my lab was still getting renovated over at WashU. And that time, that was a tough period because I wasn't doing any experiments. I was just kind of, sitting in my office trying to organize and order stuff along with my lab manager and whatnot. So I was not the happiest person because it was just like, oh, this is like administrative work, if in a way. But yeah, to me, and I kind of showed with this with somebody, but like going in the lab for me is actually kind of therapeutic as far as like just getting out of my office and doing stuff on the bench, just doing stuff I love. And so what I've done is like I have like little projects that I kind of do, or I get involved in somebody else's one of my trainees projects and kind of help them do an experiment. Um, and the cool thing about that is that they can set up all the tedious or laborious stuff, and you come in for the fun part, and you can leave, and then you can clean up afterwards. And so, yeah, it's like really therapeutic for me. So like, I still hold on to my projects, and I still have experiments I do from time to time. But I'm still trying to balance like how much time like I spend in my office doing all emails and writing and whatnot versus like bench work. And I haven't figured out that balance yet. So.
All right, thank you. Monica, you want to add something?
Sure, yeah. I'm very happy to not be in the lab. I feel like I have plenty of other things to pull me out of my office and I like have a couple mundane projects of like just organizing strains and stuff that I need that gets me into the lab. Um so it's yeah, I kind of make a point to like go hang out in the lab or like maybe hang out with my students in the lab, but I'm very happy to not be doing bench work anymore. I think it's very possible. So
Yeah, that happens a lot. I would say that it depends also on like the personality and how like how how much love do you have to certain type of experiments also because we prefer some experiments than others, so it's also different. So we go into the next question. Sunil wants some advices in how to balance your startup, how like administrate your starting up money. Monica, you have any recommendations on this?
oh, so this is going to be country specific. So in Canada, we get like 200 grand cash right away as startup. But then you have to apply. So it was interesting hearing you guys talking about like hiring people and then applying for grants, whereas here it's very much like apply for grants and then do everything else. Because some of those grants are essentially part of the startup. And there's like a very high chance that you'll get them. It's not 100%, but I did get them. So basically, what I did was I spent my entire startup on equipment. And then I just got my equipment grant. So now I can refund my startup from my equipment grant. Um, and yeah, going back to the question about fear, the thing that I am most terrified about is money and like not managing my money well and like not having money to do the things that I've already set in motion. And I've spent a lot of time worrying about that, and I've just sort of learned to like let go and trust the process and I don't know. Like do what you need to do to get the science done. And I have learned that there are sort of like slush pots of money that the department and the university has, like in case of a rainy day, in case you do really screw up. But like it's not the end of the world, which is also kind of terrifies me. Like I don't want to have to, I don't know what are those pots of money. I don't know. Um I don't know if I've really figured it out yet, but that's my experience so far. I'd love to hear what Rachel and Kevin have to say.
Yeah, this is a tough one because in my personal life, I'm a very frugal person, and it's kind of interesting to take your personal experience with money and then all of a sudden be responsible for professional money. That also includes hiring other people, right, and paying their salaries. So I don't know that I have the best answers to this, but maybe one thing that I would say is at the very beginning, right, when you have your startup and you have a shiny new lab that's empty and you're first in your office and you don't know anything and you're excited, all the reps are going to show up. All the company reps are going to show up and they're going to show you the latest, shiniest equipment, the newest, coolest stuff. And you're going to be so tempted to buy it because you have this money and oh my God, amazing. Wouldn't it be cool? So cool to have this. We might use it. But I fortunately very quickly learned that I was going to inherit a lot of equipment and some of it might be old, but it still works. It's great equipment. I did not need all the new and shiny things. So I would say at this point, I focused on trying to budget for people. That's the most important thing to me, and figuring out how I could share equipment or what equipment do I really absolutely need, what equipment already exists. So trying to think about balancing it like that. And moving forward, who knows what's going to happen, but that's the first thing that I realize I should be thinking carefully about.
Yeah, I also echo Rachel's point. When you open up a new lab, the vendors they come after you like sharks, and it's kind of funny and it's kind of annoying like at the same time because it's just like okay, like yes, I get it. Um, but yeah, as far as like whether to spin it out or to spread it out, I guess it depends on. So one factor it depends on is like some universities or institutions they let you have a startup like indefinitely, so you can keep rolling it over like every year. And then some institutions you only have, let's say, 4 years to spend your startup before they take it back. So in that case, in the latter case, then yes, it makes you more advantageous to spend your startup before the university or the institution they take it back. But in the former case, to where like you have indefinite time to keep rolling over your startup, for me, I've kind of been more, more conservative with spending, just to kind of save the startup as like a rainy day fund. You should spend it because that's what your startup is there for. So definitely spend it. But I've been, I guess, a little bit more careful as far as like, hey, we can spend on this, but we don't need a shiny bells and whistles. The basic core model is fine. So
All right, thank you. I think that we have time for one last question, and I'm gonna choose the one that is related to interdisciplinary, like the faculty position, because the others are like more funding and recruiting people. We're gonna make sure that the panelists they answer these questions, and we're gonna post them maybe later in the post. But the question is how to navigate pursuing an interdisciplinary faculty position, which is like how do you manage being there and also trying to do something more applied? Maybe Kevin, you like to start,
yeah, I'm trying to think.
or anyone that has an idea, or Rachel, Monica, feel free, please.
Yeah, it's a good question. So if anybody else want to jump in, go for it.
Yeah, for me, so I kind of, it's like my research program spans from genes to ecosystems is my tagline, and I'm sort of focusing on the genes and wet lab work first as I'm building the relationships to do more like applied field work and whatnot here. So I feel like those relationships take time to build, and in the
Mm,
meantime, doing my low-hanging fruit in the lab. So I'm also joint appointed between two different colleges within UBC. And I haven't really figured out how to balance that. I just sort of try to show up to things in both departments and TBD. Yeah.
Rachel, you have any
Yeah,
advice?
sure. Just from my personal perspective, so I'm a developmental biologist. I said that at the beginning, but I've always had some sort of systems biology component to what I was working on. So even from very early on in my PhD days, I had computational collaborators. So I was working with computational biologists or mathematicians. I'm still doing that. So I would say I'm focusing now on my strengths on the developmental biology, on kind of, as Monica was saying, really the core questions that I want to get at, while maintaining collaborations that I already have, that I already established as a postdoc. And of course, trying to participate, right, and meet new people, get out there, talk about myself, even though it's scary, talk about my stuff, meet new people, and try to build new relationships. I think that in science now, to me, I feel like for me personally, I won't say for everybody, it's impossible for me to be a jack of all trades and feel happy about how I'm doing each of them, right? I don't feel that I can be a hardcore computational person and a hardcore developmental biologist. That's just me. Maybe some people can, but I'm not sure that that's my personal identity. So I'm trying to focus on what's my expertise and think about collaborations moving forward.
Yeah. Kevin, you have one last thing to add?
No, I pretty much echo what Rachel and Monica said.
Okay. All right. Thank you. So I think that we are right on time.